Hi, my name is Sasha Thalluri, and I'm an undergraduate research assistant in Dr. Brittany Lassane's lab at the University of Alabama at Birmingham School of Medicine. Today, I'll be presenting on the characterization and prediction of DNA methylation instability across human cancers. DNA methylation refers to the epigenetic modifications that occur on the genome. When these modifications exhibit widespread altered patterns, DNA methylation instability will occur. This is an enabling characteristic of cancer. These patterns are molecular signatures that are linked to gross genomic alterations that represent distinct etiologies and are associated with drug efficacy and patient prognosis. And that's why it's so important to study DNA methylation instability and characterize DNA methylation instability phenotypes. Using this knowledge, we can further understand its role in tumorigenesis. Many studies have characterized the CpG island methylator phenotype, which refers to the methylation occurring at CpG islands. However, fewer have studied methylation occurring at non-CpG island sites. This includes CpG shore, CpG shelf, and CpG open sea sites. A CpG island is determined by its DNA sequence, which considers the number of CpG dinucleotides within a 500 base pair window. CpG shores are located about 0 to 2 kilobases away from the CpG islands, and CpG shelves are located around 2 to 4 kilobases away from the CpG islands. And so these annotations are very near CpG islands, whereas open sea sites are farther away from CpG islands and occur at different sites throughout the genome. To study DNA methylation instability, we obtained Illumina Infinium human methylation 450 bead chip array data from over 11,000 patients across 33 cancers publicly available from the Cancer Genome Atlas database, and calculated DNA methylation instability using the CpG annotations I mentioned earlier. And what's nice about TCGA is that there are matched normal and tumor samples from the same patient, so we can compare between sample types when studying these genomic alterations. Our study, in particular, identifies ways to calculate methylation patterns while comparing and contrasting them across the 33 cancers found in the TCGA cohort. To do that, we studied the global patterns of DNA methylation instability by creating specific metrics in regards to the CpG annotations. We use metrics because they provide us with a numerical representation of DNA methylation instability by providing us with a specific score. And there are two types of metrics we developed. One type considered all 450,000 probes throughout the array, and the other considered the CpG probes that exhibited the most variation, which were in the top quartile of standard deviation. The reason we wanted to also study these probes that are the most variable is because they calculate DNA methylation instability because they may highlight probes associated with the most biological variability in patient samples. And after determining probe inclusion, we calculate the mean and median to determine these global patterns of methylation seen throughout the epigenome. And first, we compared DNA methylation instability metrics to one another. Figure 3 has two plots, which show the Spearman's correlation between the average and median DNA methylation in all of the probes and in the probes that are the most variable in CpG islands. Figure 3a, in particular, shows that there are differences between the average and median. This suggests that outliers may be driving average methylation. As such, we continue to study the distributions of DNA methylation using these metrics. What we have found is that the distribution of DNA methylation between CpG annotations varies, but was consistent between clear cell renal cell carcinoma and breast invasive carcinoma, otherwise known as KIRC and BRCA, which are the two cancers we selected from a preliminary study. Specifically, we can see these differences in distribution between CpG island and CpG shore, top quartile median metrics. However, methylation across annotations by KIRC tumor stage vary, suggesting a potential association with clinical features. So we then compare the DNA methylation instability metrics with clinical phenotypes, including tumor stage. Figure 5 shows the distributions of the CpG island, shore, shelf, and open sea methylation using the top quartile median metrics. And we have confirmed that there is differing distributions based on CpG annotation. This suggests DNA methylation annotations capture clinically associated signatures. And currently, we are formalizing these DNA methylation instability metrics across these annotations and assessing them across cancers. Then we'll develop an R package to host these metrics to enable analysis in the community. Afterwards, we'll examine DNA methylation instability as a drug response biomarker within and across cancer types. And so this concludes my presentation. Thank you for watching it. I would like to thank my lab for all of their help throughout this study. If there are any questions, comments, or feedback, please let me know, and I've left my contact information on this slide. Thank you.